So today we will discuss about another food storage polysaccharide, that is glycogen. This glycogen, as I have told you earlier, it is known as animal starch. Why it is known as animal starch? Glycogen is known as animal starch. Why it is known as animal starch? Because it is the stored carbohydrate of animals, right? I'm getting some noise, please. Okay. So glycogen is animal starch because it is the stored carbohydrates of animals. So what is this glycogen? This glycogen, it is a dendrimer of. It is a dendrimer of 30,000 to 60,000. Glycogen is a dendrimer of 30,000 to 60,000 glucose residues. Glucose residues. Branching after every 8 to 12 glucose residues with 13 glucose residues. 13 glucose residues per branch. 13 glucose residues per branch. So glycogen is a dendrimer of. What do you understand by dendrimer? Dendrimer means a repeatedly branching molecule. Dendrimer means a repeatedly branching molecule. Any molecule which shows a repeated branching, that branching molecule is known as a dendrimer. So in glycogen there is a repeated branching, right? Branching is repeated very frequently. So it is a dendrimer of 30,000 to 60,000 glucose residues. So it is formed of 30,000 to 60,000 glucose residues. These glucose residues, they show branching after every 8 to 12 glucose residues. So as I told you, branching in amylopectin. We have discussed starch. In starch, there was one component, amylopectin. In that amylopectin, branching occurred. Their branching occurred after 20 to 30 glucose residues, right? Here, branching occurs after 8 to 12 glucose residues with 13 glucose residues per branch. In a single branch, there may be 13 glucose residues, right? So these glucose residues, which are present in the main chain, they are linked by alpha 1,4 glycosidic linkage, like amylopectin. Like amylopectin, these glucose residues, which are present in the main chain, they are linked by alpha 1,4 glycosidic linkage. However, at the branch point, like amylopectin, at the branch point, there is alpha 1,6 glycosidic linkage. So it is just like amylopectin. Glycogen is just like amylopectin. However, amylopectin is a very large molecule that was formed of 2,000 to 2 lakh glucose residue, right? As compared to amylopectin, it is comparatively small. It is just formed of 30,000 to 60,000 glucose residues, right? In amylopectin, branching occurred after 20 to 30 glucose residues, and there were some 20 to 24 glucose residues per branch. In glycogen, branching occurs after 8 to 12 glucose residues. Here the branch occurs after 8 to 12 glucose residues. This is one glucose residue, another glucose residue, another glucose, 4th, 5th, 6th, 7th, 8th, 9th, 10th, 11th, 12th. So in between 8 to 12 glucose residues, a branch occurs, branch develops. This is one branch, right? If we move ahead, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, another branch develops, right? So here branching occurs. Branching occurs after 8 to 12 glucose residues. And how many glucose residues are present in a single branch? In a single branch, there are 13 glucose residues, right? So it is a branching molecule like amylopectin. But the difference is that amylopectin is very large molecule, formed of 2,000 to 2 lakh glucose residues. As compared to amylopectin, it is smaller. It is formed of just 30,000 to 60,000 glucose residues. In amylopectin, branching occurred after 20 to 30 glucose residues. In glycogen, branching occurs after 8 to 12 glucose residues, right? In amylopectin, there were 20 to 24 glucose residues per branch. In glycogen, there are just 13 glucose residues per branch. The main glucose residues, they are linked by alpha 1,4 glycosidic linkage, like in amylopectin, and at branch point, like amylopectin, there is again alpha 1,6 glycosidic linkage, right? So this is glycogen. Where this glycogen is stored in animals? Where glycogen is stored in animals? It is stored in liver and skeletal muscles. So in animals, glycogen is stored in liver and skeletal muscles. In skeletal muscles. So it serves as a reserve of food in our body. It is stored in liver and skeletal muscles. Just after having a meal, an adult person may store some 100 to 120 grams glucose in his liver. Just after having a meal, right? An adult person may store some 100 to 120 gram glucose in his liver, right? So it is mostly stored in liver and skeletal muscles. Liver has a little storage capacity, while as skeletal muscles, they store a lot of glycogen, right? So this glycogen, for this reason, it is known as animal starch, because it is stored in our liver and skeletal muscles, and it acts as a reserve food whenever we fall short of glucose in blood, when our blood glucose levels will decrease. This glycogen, it will be hydrolyzed. When glycogen is hydrolyzed, what does it release? When glycogen is hydrolyzed, what does it release? On hydrolysis, in our body, it releases glucose. It releases glucose. So it maintains then our blood glucose level, right? So when our blood glucose level falls down, at that time, glycogen is hydrolyzed. On hydrolysis, it releases glucose, and our blood glucose levels are maintained. So that is all about glycogen. So there were two food storage polysaccharides, starch and glycogen. Starch is the stored carbohydrate of animals, whereas glycogen is the stored carbohydrate of, starch is the stored carbohydrate of plants, whereas glycogen is the stored carbohydrate of animals, right? So that is all about glycogen, right? Now we are going to discuss about. Now we will discuss structural polysaccharides. Is starch a homopolysaccharide or heteropolysaccharide? Or starch is homopolysaccharides or heteropolysaccharides? Polysaccharides. Is starch a homopolysaccharide or heteropolysaccharide? Starch is a homopolysaccharide because it is formed of only glucose, right? Similarly, glycogen is also a homopolysaccharide because its basic unit is glucose, right? So those polysaccharides which have got same type of basic unit, that is known as a homopolysaccharide. Now we will discuss structural polysaccharides. Structural polysaccharides. Structural polysaccharides are those polysaccharides which are involved in formation of some body structures in plants and animals. For example, in plants, we have got cellulose. You know cellulose, it is involved in formation of cell walls, right? Then similarly, we have got chitin in animals. Chitin, it is found in exoskeleton of arthropods. It is found in the cell walls of some fungi and algae, right? Similarly, then we have got mucopolysaccharides. Mucopolysaccharides. These mucopolysaccharides, they are also structural polysaccharides. So there were two food storage polysaccharides. starch and glycogen. Now we have got three structural polysaccharides: cellulose, chitin, and mucopolysaccharides. So first we will see what is cellulose, how the cellulose is formed, right? So we will first discuss cellulose as a structural polysaccharide. You know cellulose, it is the main component of cell walls of plants, right? This cellulose, it is produced by plants. This cellulose is the most abundant organic compound in the biosphere. Cellulose is the most abundant organic compound in the biosphere. It is produced by plants. This cellulose, it is a polymer of, beta glucose residues. Glucose residues. Cellulose is a polymer of beta glucose residues, right? So it is a homopolysaccharide. So its basic unit is beta glucose. In starch and glycogen, there was alpha glucose as the basic unit. In starch and glycogen, alpha glucose was the basic unit. But in cellulose, there is again glucose, but this glucose is beta glucose, right? So one beta glucose molecule. This is one beta glucose molecule. One beta glucose molecule. It combines with another beta glucose molecule. What is the difference between alpha glucose and beta glucose? What is the difference between alpha glucose and beta glucose? Come on, tell me. This is glucose, right? In alpha glucose, this OH group, it is downwards. In beta glucose, this OH group is upwards, right? In alpha glucose, OH group is lying downwards, whereas in beta glucose, this OH group is upwards. Otherwise, it is same, right? So this is carbon first, carbon second, carbon third, carbon 4, carbon 5, and carbon 6. So it is a 6 carbon containing compound. This glucose is known as beta glucose, right? So in this way, one beta glucose molecule, it joins with another beta glucose molecule. This is another beta glucose molecule. One beta glucose molecule joins with another beta glucose molecule. This is another beta glucose molecule, right? So when they join by the formation of a bond. Which bond will be formed in between them? A molecule of water will be eliminated like this. A molecule of water will be eliminated. This water molecule is eliminated in between them. So between these two beta glucose units, a bond is formed. Which bond will be formed here? When a sugar joins with another sugar, what type of bond is formed? A glycosidic bond is formed. A glycosidic bond is formed, right? So by the elimination of a water molecule, this water molecule it is eliminated, and a glycosidic bond is formed between these two beta glucose sugars, right? A glycosidic bond is formed between these two beta sugars, right? Similarly, another glucose molecule will join. Another glucose molecule will join. Another glucose molecule will join. Similarly, in the same way, there may be hundreds to thousands of glucose residues. Thousands of glucose residues in a single polysaccharide chain. So in the same pattern, in the same manner, one glucose, another glucose, another glucose, another glucose, another glucose. How many glucose residues can join? Some hundreds to thousands. The number is not specific. So hundreds to thousands of glucose residues may join, and they may form a long polysaccharide chain. a long polysaccharide chain is formed, right? A long polysaccharide chain is formed. So in this polysaccharide chain, the beta glucose units, they are linked by a glycosidic bond like this. They are linked by a glycosidic bond like this. This is oxygen. This is the glycosidic bond, right? So they are linked by glycosidic bond. They are linked by glycosidic bond, right? What will be this linkage known as? It is between carbon first, this is carbon second, this is carbon third, this is carbon fourth, this is carbon five, this is carbon three. So what this linkage will be known as? This linkage is between carbon first and carbon four, right? What will be named as? It is named as beta 1,4 glycosidic linkage. It is named as beta 1,4 glycosidic linkage, right? You know, we can digest starch. We can digest glycogen. Starch and glycogen. They also contain glucose, but they contain alpha glucose. In them, there are alpha glycosidic linkages. In starch and glycogen, there are alpha 1,4 glycosidic linkages. This is cellulose. It is also a polysaccharide, right? It is also formed of glucose, but we are not able to digest cellulose. We are not able to digest cellulose. Why we are not able to digest cellulose? Why we cannot digest cellulose? If the structure is similar in starch, in glycogen, there are glucose units, there are alpha glucose units, right? They are linked by alpha 1,4 glycosidic linkage. In cellulose, there are again glucose units, though they are linked by beta 1,4 glycosidic linkages. We can digest starch, we can digest glycogen, but we are not able to digest cellulose, right? Did you know that we are not able to digest cellulose? Did you know that we are not able to digest cellulose? We are not able to digest cellulose because in our body, a starch digesting enzyme, alpha amylase is secreted. But for the digestion of cellulose, we require beta amylase, right? In starch and glycogen, which linkage was there? In starch and glycogen, there was alpha 1,4 glycosidic linkage, right? In cellulose, there is different type of linkage. There is beta 1,4 glycosidic linkage, right? So we secrete alpha amylase. Our body produces alpha amylase. That alpha amylase can digest starch and glycogen. But this alpha amylase, it cannot digest cellulose. It cannot digest cellulose, right? So this is the reason we are not able to digest cellulose. Because in cellulose, there are beta 1,4 glycosidic linkage. In cellulose, there are beta 1,4 glycosidic linkage. Don't make noise, please. Don't make side talks, please. In starch and glycogen, there is alpha 1,4 glycosidic linkage. This linkage can be broken by alpha amylase. In cellulose, there is beta 1,4 glycosidic linkage, which cannot be broken by alpha amylase. For this purpose, we require beta amylase, which is not produced by our body. So our body doesn't secrete beta amylase. That is why we are not able to digest cellulose, right? So now you know hundreds to thousands of glucose residues. They join each other by beta 1,4 glycosidic linkage, and they form a long polysaccharide chain. They form one long polysaccharide chain. Similarly, there may be another long such chain. There may be another such a long polysaccharide chain. So these long polysaccharide chains, they come to lie parallel to each other. They come parallel to each other. And they are projecting hydroxyl groups. They are projecting hydroxyl groups. They are projecting hydroxyl groups. They form bonds with each other. The hydroxyl group, which is lying downwards on this chain, it forms a bond with the hydroxyl group, which is lying upwards on this chain, right? So hydroxyl groups, they eliminate water in between them, and bonds are formed. So these bonds, they hold these chains together. They hold these chains together. These bonds, they hold these chains together. And in this way, when many such polysaccharide chains combine, they form a bundle of polysaccharide chains known as cellulose microfiber. Cellulose microfiber, right? So in this way, cellulose microfibers are formed. You got me? What I am saying? So first, hundreds to thousands of glucose residues. They join each other by beta 1,4 glycosidic linkage. So they form a long polysaccharide chain. So first, a single polysaccharide chain is formed. Then many such polysaccharide chains. One polysaccharide chain, another polysaccharide chain, another polysaccharide chain, another polysaccharide chain. Many polysaccharide chains, they come parallel to each other. They come parallel to each other. The hydroxyl groups, which are projecting upwards or downwards, they form bonds with the neighboring polysaccharide chain. This is a single, this is one polysaccharide chain. This is another polysaccharide chain. This polysaccharide chain, it has got some hydroxyl groups downwards. This polysaccharide chain, it may have some hydroxyl groups projecting upwards. So this downward projecting hydroxyl group and upward projecting hydroxyl group, they form a bond by the elimination of water, right? So that joins the polysaccharide chains together. So in this way, many polysaccharide chains, they join with each other and they form a bundle of polysaccharide chains. That bundle of polysaccharide chains is the cellulose microfiber. Then this cellulose microfiber, it is laid in the gel-like matrix of hemicellulose and some pectins to form the cell wall, right? This, these cellulose microfibers, they are laid in the gel-like matrix of hemicellulose and pectins to form the cell wall. So that is about the cellulose. Did you understand all about the cellulose? So what is the basic unit of cellulose? What is the basic unit of cellulose? The basic unit of cellulose is beta glucose, right? So how do these beta glucose units combine? These beta glucose units, they join each other by beta 1,4 glycosidic linkage. They join by beta 1,4 glycosidic linkage. How many beta glucose residues join together to form a single polysaccharide chain? Hundreds to thousands. Sometimes, sometimes 10,000 glucose residues have been found in a single polysaccharide chain. So hundreds to thousand glucose residues, they join together and they form a single polysaccharide chain. Then similarly, another polysaccharide chain is formed. Then many polysaccharide chains, they come to lie parallel to each other. When they come parallel to each other, the hydroxyl groups which are projecting upwards or downwards between two polysaccharide chains, they form hydrogen bonds, they join each other, they form bonds, and by those bonds, these polysaccharide chains are held together in the form of a bundle. And this bundle of polysaccharide chains, it will be now known as cellulose microfibrils. Now, these cellulose microfibrils, they are laid in the gel-like matrix of hemicellulose and pectins to form the cell wall. So that is all about cellulose, right? Now, after cellulose, we have got another structure of polysaccharide. Now we have got another polysaccharide, another structural polysaccharide, that is chitin. That is chitin. This chitin, it is just like cellulose. It is just like cellulose in its composition. In its composition. Chitin is just like cellulose in its composition. But where we find chitin? This chitin, it is found in the exoskeleton now. Exoskeleton of arthropods. Arthropods. It is found in the cell walls of most of the fungi, and some algae, algae, right? So chitin, it is just like cellulose in composition. It is found in the exoskeleton of arthropods, the chief component of exoskeleton of arthropods. You know, arthropods, they are covered externally by hard chitinous plates, right? Those chitinous plates, they are mostly formed of chitin, right? And it is also found in the cell walls of most of the fungi. So fungi, they don't have cellulose usually in their cell walls. Instead of cellulose, they have chitin in their cell wall. And in some algae, there is also found this chitin is also found in cell wall, right? So as I told you, it is just like cellulose in composition. Then how does it differ from cellulose? What is the difference between cellulose and chitin, right? It differs from cellulose in its basic unit. What was the basic unit of cellulose? What was the basic unit of cellulose? Cellulose was a polymer of what? Beta glucose. Good. Exactly. Cellulose is a polymer of beta glucose units. But this chitin, it is a polymer of N-acetyl glucosamine units. NAG. It is a polymer of N-acetyl glucosamine units or residue. It is a polymer of N-acetyl glucosamine residue. So what is this N-acetyl glucosamine? We also call them as amino sugars. These are modified sugars. These are modified sugars. N-acetyl glucosamine, whose acronym is NAG, NAG. NAG, NAG. It is the acronym for N-acetyl glucosamine. N-acetyl glucosamine, right? This N-acetyl glucosamine, it is a modified sugar. It is a modified glucose. Where the modification is, I will show you. It is again 6 carbon containing compound like glucose. Here is OH group, here is H, here is H, and here is the modification. Here we have got NH, C, double bond O, CH3. So this group is known as acetyl group. This group is known as acetyl group. So it has got acetyl group, nitrogen containing group, N-acetyl group, right? So it is a sugar. It has got modification. This is carbon first, carbon second. At carbon second, it has got an acetyl group. This is carbon third. This is carbon fourth. This is carbon 5 and 6. So it is just like a glucose molecule. You can see it is just like a glucose molecule, right? But at carbon second, it has got a modification. It has got an acetyl group attached to it. That is why we call it as an N-acetyl glucosamine. That is why we call it as NAG, N-acetyl glucosamine, right? So the difference between glucose and N-acetyl glucosamine is that it has got an acetyl group at carbon second. So one N-acetyl glucosamine, it combines with another N-acetyl glucosamine. So these are modified sugars. One N-acetyl glucosamine combines with another N-acetyl glucosamine. Right? It again combines by the elimination of a water molecule. One water molecule will be eliminated. This water molecule will be lost. Right? And a bond will be formed between these two modified sugars. Which type of bond will be formed here? Which type of bond will be formed here? Again, a glycosidic bond is formed. Glycosidic bond is formed. And what will be this linkage known as? What will be this linkage known as? This linkage will be known as alpha. Sorry, not alpha, beta. Because it's a beta sugar. OH group is upwards. So this is beta N-acetyl glucosamine. Right? It's a beta sugar. This is also a beta sugar. So which type of linkage will be formed? The linkage formed here will be beta 1,4 glycosidic linkage. The linkage will be beta 1,4 glycosidic linkage, right? So in this way, one modified sugar, beta N-acetyl glucosamine, joined with another beta N-acetyl glucosamine, and many such sugars join. Again, the number may be hundreds to thousands, and they form a long polysaccharide chain like cellulose, right? Then many such polysaccharide chains, they come parallel to each other and they form the chitin molecules. They form the chitin molecules, right? So in composition, it is just like cellulose. In structure, it is just like, in composition, it is just like cellulose. But what is the difference? The basic unit of cellulose was beta glucose, and in chitin, the basic unit is beta N-acetyl glucosamine. In chitin, there are modified sugars. Modification has occurred at carbon second. At carbon second, these sugars, they possess an acetyl group. They possess an acetyl group. They possess an acetyl group, right? So that is why we call it as N-acetyl glucosamine, right? So these are modified sugars. So they form long polysaccharide chains. Long polysaccharide chains come parallel to each other. They form bonds between them, and then they form the chitin molecule. Then this chitin is deposited in the cell walls of, that is deposited in the exoskeleton of arthropods. It is found then in the cell walls of fungi and some algae. The bond will be formed like this here. Bond is formed like this, right? Bond is formed like this. So this is the chitin and cellulose. So both chitin and cellulose, they were structural polysaccharides, right? And both of them are homopolysaccharides. Why they are homopolysaccharides? Why chitin and cellulose fall in homopolysaccharides? Because the basic unit is similar, right? One nog, another nog, another nog, another nog. In cellulose, there was beta glucose, beta glucose, beta glucose. So their basic unit is similar. They are monomers of the same type. They are polymers of the same type of monomers. They are polymers of same type of monomers. So that is why we call them as homopolysaccharides, right? So this is section for today. Our time is running out. If you have any doubts, you can tell me. Okay, thank you for attending my class. Thank you.